This is a lecture video over Chapter 3 on cells, the living units, and why this material matters in this chapter. In the context of health professions, understanding the structure of a body's cells explains why the permeability of a plasma membrane can affect treatment. So knowing about the membranes of cells can affect how we design treatments for patients. In this chapter, you're going to learn that in section 3.1 that cells are the smallest unit of life, and we're going to be exploring 3 parts. In part 1, we're looking at the plasma membrane. We're going to be asking what is the structure of the plasma membrane, how do substances move across the plasma membrane, and we're going to look closer at modes of movement. In 3.3, we're going to look at passive membrane transport, and in 3.4, active membrane transport. And then in 3.5, we're going to ask how does a cell generate a voltage across its plasma membrane, which is what neurons do, nerve cells. And in 3.6, how does the plasma membrane allow the cell to interact with its environment? Moving on to part 2, we're going to examine the cytoplasm, and we're going to look closely at the components, which include the cytosol and inclusions. And in 3.7, we're going to look at the cytoplasmic organelles, and we'll further explore cellular extensions, which would be things like flagella and cilia. And then in part 3, the nucleus, we're going to be looking at investigating the structure of the nucleus and asking in 3.10, how does a cell grow and divide? And then in section 3.11, what are the roles of DNA and RNA in protein synthesis? And then we'll further ask in section 3.12, how are cells, organelles, and proteins destroyed? So, moving to section 3.1, titled The Cells, the Smallest Living Units, our learning outcomes are going to be to define cell, name and describe the composition of extracellular materials, and list 3 major regions of the generalized cell and their functions. So a major theory in biology is cell theory, and here the cell is considered to be the structural and functional unit of life. So the smallest unit that can be considered life is the cell. Now, how well an entire organism functions depends on the individual and combined activities of all of its cells, especially if you're a multicellular organism like a plant or an animal or the human animal. Another big overarching idea in biology follows through with at the cellular level, in that structure and function are complementary. And here, when we're looking at structure and function in cells, biochemical functions occurring in cells are going to be dictated by the shape of the cell and specific subcellular structures. And so what makes different cells depends on what the components are, various components are within the cell. And then another big idea from cell theory is that you cannot get cells unless you have pre-existing cells. So that's for the continuity of life, or life to continue. Only cells can arise from other cells. So you must have pre-existing cells to get other cells. So looking at cell diversity, we have over 250 cell types in the human body. So that's 250. And the types differ in size, shape, and subcellular components. And these differences lead to differences in their functions. So looking at some different groups of cells by category, in A here we have cells that connect body parts, form linings, or transport gases. So the erythrocytes, for example, which are red blood cells, transport gas, and they have this unique disc shape. They also lose their nucleus during development. We have fibroblasts, these cells right here, and they do like it sounds. These are fiber makers. You can see the protein fibers they make. And then we have epithelial cells, which line or cover structures. And then we have another group, B. These are cells that move organs and body parts. So this would be cells capable of contracting or shrinking. And here are your voluntary skeletal muscles right here. They're long because their function is to shorten or to contract. And then we have smooth muscles, which you find in hollow organs like the stomach. And then we have cells that store nutrients. And here you have an adipocyte or fat cell. And then we have cells that fight disease, and here's a macrophage, which literally translated means large eater. This type of cell could, for example, take in bacteria and kill them and digest them down. And then we have this one here, cell for communication. Here's a nerve cell. Nerve cells conduct electric impulses down their extensions here. And then we have cells for reproduction, like this sperm cell, which is really just largely a nucleus, and another major structure is the flagellum in the back to propel it towards the egg for fertilization. So here is a picture of our composite or generalized cell on the right, and you can see the components that are in there. For example, the nucleus right here. So what we want to do is become aware of the fact that all cells have some common structures, even all human cells. And number one common structure is that they have a boundary that's flexible called the plasma membrane. And then number two, they have a cytoplasm, which includes everything in there, including the cytosol and all the organelles. Everything inside the cell, all those folded membranes, nucleus, and the fluid in there, the cytosol, would be the cytoplasm. And then, at least at some point in development, they're going to have a nucleus. Even the red blood cell that loses its nucleus will has had it, at least initially had a nucleus early in the development of the blood cell. So, extracellular materials here. Extra means beyond. So this would be materials that are found outside the cell. So there we go. Substances found outside the cell. There are some classes of extracellular materials, and they include extracellular fluids. They include cellular secretions and extracellular matrix. So going back to the fluids here, these include interstitial fluids. All cells are submerged or bathed in this fluid. Plasma in the blood, that's the fluid, or the liquid part of the blood. And cerebral spinal fluid, which is fluid surrounding the nervous system organs. Some cellular secretions include things like secretions like saliva and mucus. And the extracellular matrix are going to be substances that act as a glue to hold cells together. So, the three main components of all cells include the plasma membrane, the nucleus, and the, is it A, DNA, B, cytoplasm, C, organelle, or D, cell wall? So part 1 of chapter 3 covers the plasma membrane. And the plasma membrane acts as an active barrier, separating the intracellular fluid from extracellular fluid. It's going to play a basic role in cellular activity by controlling what enters and what leaves, and we also will refer to it as the cell membrane. The learning outcomes for section 3.2, including to describe the chemical composition of the plasma membrane and relate it to the membrane functions, and then compare the structure and function of tight junctions, desmosomes, and gap junctions. The plasma membrane is going to consist of membrane lipids, and they're going to form a flexible lipid bilayer. So you can see the model on the right, you can see there appears to be two layers of molecules facing in opposite directions. The specialized membrane proteins that are in there, which are the purple structures that you see, circling one right there, they're going to flow through this fluid membrane, and this results in a constantly changing pattern in there, and we refer to this model as a fluid mosaic model. We're also going to have surface sugars that form the glycocalyx, and those are the green chains of sugars there on the surface. And some of the membrane structure is going to help to hold cells together through cell junctions. This was mentioned in the objectives earlier, things like desmosomes and gap junctions. So looking at the chemical composition of what we see in there, the bilayer is going to be formed by these phospholipids, and they're going to have hydrophobic tails preventing water-soluble substances from crossing through the boundary there of the membrane. So you can see the basic molecule here is a polar head, which is hydrophilic, and a non-polar tail, which doesn't like water. And then on the image on the bottom here, you can see the two layers of those forming the major portion of the membrane. Then we also have these yellow molecules, four-ring structures. And this is cholesterol, and cholesterol helps to stabilize the membrane. It helps to keep the membrane from being too fluid. And then it also, because it's a lipid, it helps also decrease water solubility of the membrane. So that helps to prevent things from just moving in and out of the membrane. And then we have proteins. Those are the purple structures in our model drawing below. And they help determine what functions the membrane can perform. There's many roles. For example, proteins involved in transporting things across the membrane, communication, where they act as receptors for things like hormones, the signal molecules, and joining cells to each other and to the extracellular matrix. And then we see that proteins are going to have different shapes, and the shape, remember, the structure determines the function, so the shape of these proteins is important. Carbohydrates, they're going to act as identity molecules, and they allow cells to recognize who's who. For example, during development, cells can sort themselves into tissues, so cells recognize each other as they're moving into appropriate positions during fetal development and embryonic development. They also allow immune cells to recognize self from non-self, our own cells from pathogens, and they're found only on the outer surface of the membrane. And this kind of compares it to like the sugar coating on cereal, breakfast cereal. Together, the carbohydrates on the outside of the cell are referred to as the glycocalyx, which literally translates to sugar coating, covering. So looking at the description here, we have our phospholipids that form the phospholipid bilayer, and they're capable of moving around side to side and rotating, as these arrows suggest, but flipping from one side to the other is difficult to do for an individual phospholipid. And then the yellow four-ringed molecules, those are the cholesterol, and those can move easily between the two layers. And then we have integral proteins, and those are going to be embedded in the lipid bilayer. And then peripheral proteins, those are going to be anchored to the membrane or to other proteins. So we can see here there's a peripheral protein, and then it's got this anchoring lipid-like molecule that helps keep it in there. And some may be anchored to proteins themselves. Some go all the way through. Here's a peripheral protein there on the top that's anchored to a integral protein. Then we have carbohydrates at the surface, and they can be attached to proteins, and we call those glycoproteins for sugar proteins. And there's your chain on a protein there, on an integral protein. And then we have carbohydrates that can be attached to lipids, and we'll call those glycolipids. So there's one there called a glycolipid. Looking at the other model we saw earlier, just kind of reviews the basic functions of the plasma membrane overall due to its structure. First of all, it's a physical barrier, and it encloses the cell, separating the cytoplasm from the extracellular fluid. It is selectively permeable, or they say it's selective permeability, because it determines what substances enter or exit the cell, so not anything can go through. In fact, at the middle of the layer is hydrophobic, or It doesn't allow things that are hydrophilic, both on the outside and the inside of the cell, to just cross easily. So things in the extracellular fluid outside and intracellular fluid are regulated as far as what gets through. And then the plasma membrane also has involved in communication. The plasma membrane proteins will interact with specific chemical messengers and relay messages to the cell's interior. And then cell recognition, the surface carbohydrates allow cells to recognize each other. So looking at membrane lipids themselves, it's a lipid bilayers, so 75% is going to be phospholipids, and you see the model on the right. You got your polar head and your nonpolar tails there. The heads are referred to as hydrophilic, which means loving water, and the fatty acid tails are nonpolar, and that means they're hydrophobic or water-fearing. 5% of the lipids are going to be glycolipids, which are going to have those sugars emerging out at the surface. And then 20%, but that can be variable, but about 20% cholesterol, and that helps improve membrane stability. Overall, you can see this model here on the right. Here, a kind of schematic here with the intracellular fluid on the inside, and on the outside would be extracellular fluid here. Our polar heads are going to face the water inside and outside the cell, and the nonpolar tails are going to be hiding here in the middle of the membrane. And then we have membrane proteins. Back to that slide. This is going to allow cells to communicate with the environment. They make up about half of the mass of the plasma membrane, even though the lipids make up 75% of the membrane. Most have specialized membrane functions. Some float freely and some are going to be tethered to intracellular structures. There's going to be two types, the integral proteins and the peripheral proteins, which we've already mentioned. With the integral proteins, they're firmly inserted in the membrane. Most are going to be transmembrane proteins. So this is just a specific kind, and that means they go all the way across the membrane. So you see most of them are transmembrane, going from one side to the other, like we do see over here. Others only go partial way, so in this case that would just be an integral protein. Now they're going to have both hydrophobic and hydrophilic regions. This is the only way they can exist within the membrane. Okay, so the hydrophobic areas are going to interact with the lipid tails. So if I were to shade in right here, or just kind of draw here, this region from here to here, all of that would have to be hydrophobic in order to be in the area where the lipid tails are, because that's hydrophobic there. And then the hydrophilic areas would interact with the water. So toward the outside here where the polar heads are, out here on the outside here, that would be hydrophilic areas, or polar areas of the protein. Now, what do these functions, these integral proteins do? They're going to function in things like transport, and they can form channels or carriers. We'll distinguish those in a little bit. Some will work as enzymes, helping reactions to occur in your metabolism, and some will act as receptors for messenger molecules. Then we have peripheral proteins, and these are going to be loosely attached, some to the integral proteins. Some will be loosely attached to the bilayer itself, and they include filaments on an intracellular surface used to support the plasma membrane. So we can see some of the parts of what we might call the cytoskeleton here, and there's a filament protein connecting to a peripheral protein there. Some of the functions of these peripheral proteins include enzymes. Some are motor proteins for shape changing during cell division and muscle contraction, which we'll study later to see how the muscle works. And for cell-to-cell connections are some of the functions for peripheral proteins. Overall, this is a diagram in your textbook. We're going to look at each individual image, which describes a basic function that these membrane proteins can perform. So here's one on transport here, and you can see the one on the left here. We have a specific transport protein that allows molecules to go through. This might be a sodium channel, for example, allowing sodium ions to go through. But the inside here of this protein would be hydrophilic, or out here this would be, the outside would be hydrophobic, so the protein has to be embedded in the membrane, but inside here that channel would be hydrophilic, allowing these hydrophilic particles to go through. Others, like the one on the right, forms a actively transporting membrane, and this would require energy to move particles through the membrane. And usually this is moving particles from lower concentration to higher concentration, which is an active process that we'll see later. And then we have some that act as receptor proteins, and that's to transduce information inside, or to pass that information along. So that's called signal transduction. So here might be like a hormone molecule, and here you have a receptor that has a very specific shape for that chemical messenger molecule. When it binds there, it will change the shape of the protein, and that will send the shape change actually will cause a series of other changes to occur within the cell. So there the signal would have been transduced, and that basically changes the metabolic activity of the cell. And then we have some that serve as enzymes. So here you can see there is a series of, in this simple diagram, are set up two proteins there that create what's called a metabolic pathway here. And so what happens here is a substrate will come in and go through a change due to this enzyme, and the next enzyme is ready to receive the product of that reaction, and then turns that product, or what we call a chemical intermediate, into the next product, and so on. So this arrangement here would assist and help prevent intermediates from going through unwanted reactions. So this will help control the process in a pathway there. And then there's cell-to-cell recognition here. And here you can see in the image here, there's a glycoprotein there on one membrane sticking out its carbohydrate chain, and the cell on the other side has another protein there. And then there's a sort of recognition for the little signaling molecule there. And so some glycoproteins, which are going to serve as identification tags that are specifically recognized by other cells. So you can see the cell on the top is recognizing the cell on the bottom in that way. And then we have proteins that are involved in attachment to the cytoskeleton as well as the extracellular matrix. You see the protein here. The membrane protein there is connected to the cytoskeleton here and below, and some of the protein fibers that are part of the extracellular matrix there. So they're going to help do things like perhaps anchor integral protein in the membrane. They're also going to help maintain shape and fix the location of certain membrane proteins, as I said, anchoring. And they may play a role in cell movement. Then we have cell-to-cell joining in here. You can see they have the word chem, which is defined here in the text. And you can see there's cell number 1 here and cell number 2 below, the two plasma membranes, and you can see the two membrane proteins interacting. So let's see what the caption here says. The membrane proteins of the joining or adjacent cells may be hooked together to form various intracellular junctions, which we'll cover here, and they include things like desmosomes, gap junctions, and tight junctions. The CAMS itself stands for cell adhesion molecules, and this provides temporary binding sites for cells as they migrate and other cell-to-cell interactions. So sometimes cells have to move past each other, and these CAMs may assist in that process. Then we have the glycocalyx here, which we've mentioned before already, and it's with those the overall sugar covering there. They stick out at the surface, and we know that some sugars are going to be attached to lipids. We call those glycolipids, and some are going to be attached to proteins. We call those glycoproteins. It's a little bit redundant. We've already mentioned this before. Now, every cell type, this is a little bit new, is going to have a different pattern of those sugar molecules. Okay, so Basically, over 200 different cell types will have different sort of a different signature on there, based on the differences of that in that glycocalyx. And so those will function as specific biological markers for that cell. Different cells recognizing each other, and this also allows the overall glycocalyx allows us to recognize self from non-self, anything that got in the body that belonged there. Looking at a homeostatic imbalance 3.1, this clinical study information here with regards to cancer cells in the glycocalyx. When cancers form or cancer cells form, that can change the glycocalyx rapidly, and the immune system can't recognize the cells as being damaged. And this way, the immune system can't dispose of the cells, so the mutated cell is not going to be destroyed and can continue to divide, potentially spreading the cancer. We're going to look at cell junctions here. Now, some cells are just going to be free. They're not going to be bound to any other cells, and they include things like blood cells, which are just free there within the plasma, or sperm cells that are just free and not bound to anything. But most cells are going to be bound to form tissues and organs. And there's three ways that we're going to look at that they can bind to each other. They include tight junctions, desmosomes, and gap junctions. So looking at tight junctions here, and overall, the diagram here on the right shows a layer of cells. This is probably, if I had to guess, this would be the inside of the small intestine right here. So this would be the lumen, or the space inside the tubular small intestine. And you can see the microvilli there that improve surface area here. And then down here is the base membrane that the cells that absorb nutrients would go in this way through the cell. As you can see that all along here, those chains of molecules, those are forming tight junctions between the cells. And so that's going to do an important function, and that's going to help to prevent things from just going right through and in between the cells. So let's take a look at what it says here. It says that these tight junctions are formed from integral proteins on adjacent cells. They're going to help to fuse and form an impermeable junction that encircles the whole cell. As you can see, they took the image right here and are blowing it up. See how those integral proteins help to hold the membranes tightly together. Now this is going to prevent fluids and most molecules from moving in between, as I mentioned before. And so where in the body might this be useful? This is going to be useful basically where we have surfaces on organs, and that's going to help just anything from getting into circulation. We need to be able to regulate. So in this case, in the case of digestive system here, this would allow the cells themselves to regulate what gets into the cell and then passes on to the other side to get into the bloodstream. And then we have desmosomes, and again, it's the same image there, but we're going to be looking at a different area here. So we're going to zoom in right here to the image there, and this would be our desmosome right here. And desmosomes are kind of like rivets, and they're going to be formed when linker proteins called cadherins, and those cadherins are those blue proteins in there, and they're going to form interlocking, kind of like the teeth on a zipper. These linker protein is then going to be anchored to its cell through a thickened button-like structure, and we're going to call those things plaques. So you can see there's a plaque right there, this yellow structure right here. And then there's going to be keratin filaments that connect the plaques intracellularly for added anchoring. So these keratin proteins are part of the intermediate filaments of the cytoskeleton. So you're literally attached to the skeleton of the cell, and that forms a stronger but flexible connection between the cells. The tight junctions are best basically holding on to the plasma membrane, which is very fluid, so we need something a little stronger here. So these desmosomes do give a little bit, and that's going to help prevent tearing, but still provide a rather strong connection between cells. So where might these be useful? Anywhere tissues have stress, where tissues are stretched, so that can create some stress on the cells. And then we have gap junctions. And gap junctions are going to form gaps. So in the image here, we're going to look at a different portion of the cell and then zoom in there. And there you can see there's these proteins that go across both membranes of adjacent cells. And they're formed by proteins called connexons that have a sort of a hexagonal arrangement. But going right through them is a gap, and that's going to allow very tiny materials to go through. So again, the name of the proteins, or transmembrane proteins, are called connexons. And what this is going to do is going to allow for cells to communicate with each other by allowing the spread of ions and nutrients like simple sugars and other small molecules between there. In cases where cells communicate by electrical signaling, like heart muscle and smooth muscle, this is going to allow for electrical signals to be passed quickly, stimulating one cell and one right near it right away. So this again is true of cardiac and smooth muscle. Skeletal muscle doesn't have these gap junctions, but we'll study that structure and physiology of muscle later. So many substances though must move constantly across the membrane, and some molecules can pass through easily and some do not. Remember that the cell membrane is referred to as selectively permeable, and it only allows certain molecules to cross. And there's two essential ways that substances cross the plasma membrane when they do. One is passive transport, which we'll study in another subsection. And it's passive because it requires no energy. And then active transport, and that's going to require energy. And the way we think about this with a physical analogy here, anytime substances have to cross your membrane and they're going from a high to low, that would be passive. It's like going downhill, like a rock moving downhill. But anytime substances have to go from high concentration to low concentration, that would be active. So this would be passive process where molecules move from high to low concentration, and then this would be active. So it's a good way to remember it. So we were to see, if I were to model my plasma membrane with two lines right here, and on the right side I had a bunch of a certain particle, and they could get through the membrane. Let's say these are sodium ions, and then there's very few of those sodium ions on here, but then we had a sodium channel right here that allows sodium to move. The direction that this would move passively would be from going from the right side to the left. That would be passive across the membrane. Now if the sodium ions were to be moved to the other side, to the right, then that would be going uphill, and that would require energy. So to move any of these this way that would be active in this case, because that's not the natural way. And moving in this drawing to the left would be passive because that's going downhill. So looking at a couple of practice questions here. Phospholipids orient themselves in aqueous solutions such that A, the polar heads are pointed toward the center of the membrane. B, the nonpolar tails are oriented toward the interior of the cell next to the cytoplasm. Or C, the polar heads and nonpolar tails alternate, facing inwards. Or D, the polar heads face the interior and exterior of the cell, with the tails forming the center of the membrane. Next practice question. In areas of the body subject to high degree of mechanical stress, which of the following types of membrane junctions would you expect to be most prevalent? Is it A, desmosomes, B, microvilli, C, tight junctions, or D, gap junctions? In section 3.3, we're going to be dealing with passive membrane transport. Your learning outcomes here are to relate the plasma membrane structure to passive transport processes and to compare and contrast simple diffusion, facilitated diffusion, and osmosis relative to substances transported, the direction that they're transported, and the mechanism of passive transport. Passive transport is called passive because it requires no energy. There's going to be three types of passive transport. The first is simple diffusion, then there is facilitated diffusion, and then osmosis. All types involve diffusion. which is going to be the natural movement of molecules from areas of high concentration to areas of low concentration. And in order to do this, there has to be a gradient, a concentration gradient. So this may also be referred to as moving down a concentration gradient. So you'd have an area where there's higher concentration of particles, and then an area where there's lesser concentration. So overall, the net movement would be in the direction from high to low. So when it comes to this diffusion, we have to understand that all molecules have a random high-speed movement due to their kinetic energy. Kinetic theory states that all the particles are consistently moving or constantly moving as long as temperature is above absolute zero. Now, when molecules are moving around and free to move, they're going to be colliding with each other. And the molecules in higher concentration areas are going to collide more, and this results in them being scattered to areas of lower concentration. So this is modeled here with a dye pellet on the bottom of this beaker here. And so as the dye begins to dissolve away, there's going to be a higher concentration of dye particles here, and as they collide with each other, they'll begin to spread outward or away from the concentration there. Now more particles will be moving away than coming back, so overall there'll be a net movement away. This is going to continue, you see here in the second picture here, and we're still going to have some moving back in the opposite direction, but overall there's more movement away, so there's still a net movement away from where the pellet was. And now that the pellet is completely dissolved and the particles are evenly distributed, if we just look where the pellet used to be right here, we're going to see that any particles moving away are going to be matched by the dye particles moving back. So now there is no net movement in any given direction. They're just, all of the dye particles are moving around randomly, and we're going to call that now, once there's no net movement, we'll say that the system is now in equilibrium. So the speed of diffusion is influenced by three factors. One is the concentration, and so there's a greater concentration, there'll be more collisions there, so that would result in a faster diffusion. And then molecular size, we know that at the same temperature, smaller molecules will be moving faster in order to have the same average kinetic energy. So small molecules diffuse faster. And then temperature, as temperature goes up, we know the average kinetic energy of the particles and the speed of the particles increases. So again, more collisions, more scattering, more diffusion or net movement. Again, equilibrium will be reached when there is no net movement of molecules in any direction. So that's the case when the particles are evenly distributed. Molecules do have a natural drive to diffuse down concentration gradients that exist between extracellular and intracellular areas. So now we're going to apply the idea of diffusion occurring across the plasma membrane from either from outside in or inside out, depends on where the higher concentration is. When we look at non-polar, which are hydrophobic, the non-polar lipid core of the plasma membrane, remember that the center of the membrane has the non-polar tails there. The non-polar is there in the middle of the plasma membrane, right here. That's going to basically stop the diffusion if there is a concentration gradient, and that makes the membrane selectively permeable. This is because most things that are in the extracellular fluid outside and the intracellular fluid inside are dissolved in a polar environment. So those substances are hydrophilic, so they cannot easily cross this non-polar area of the lipid bilayer. And so this is what makes the plasma membrane differentially permeable or selectively permeable, because those things can't simply go right through. Now, there are some molecules though that can simply diffuse through the membrane. and they include lipid-soluble and nonpolar substances. They can go through that nonpolar center of the plasma membrane. Also, some very small molecules can pass through the membrane or through some channels that may be there. So this would be referred to as simple diffusion when the movement is right through the bilayer. Now, if we have larger or non-lipid soluble or polar molecules, they can cross the membrane, but only with the help of proteins or carrier molecules. And these are going to be referred to as facilitated diffusion. And then there's osmosis, and this is a special name for the movement of the solvent, which is usually water. And so it's like diffusion of basically water for our system, or whatever solvent we might be talking about. But in living systems, usually water is the solvent. And I think this statement would be better written if it said, instead of not molecules, we would write solutes, or anything dissolved in the solvent. So if we reread it again, osmosis is the special name for movement of the solvent, not solutes. Whereas the diffusion, whether it's facilitated or simple diffusion, that would be the solutes moving. So looking at homeostatic imbalance 3.2 in a clinical setting, if the plasma membrane is severely damaged, then substances would freely diffuse or move in and out of the cell, and that would mess up the concentration gradients that are necessary for a normal functioning cell. As an example, burn patients would lose their fluids because the membranes are damaged. So fluids, proteins, and ions would be lost. So a major concern immediately would be the loss of fluids, dehydration, and loss of electrolytes. Those are immediate, life-threatening for severe burn patients. So looking at simple diffusion now, which was mentioned here a while ago, nonpolar lipid, these are nonpolar lipid soluble, which would also be hydrophobic substances, can diffuse directly through the phospholipid bilayer. Examples include oxygen, carbon dioxide, steroid hormones like estrogen and testosterone, and fatty acids. So those things can diffuse right through the membrane because they're lipid soluble, and they can make it through that middle of the membrane that is non-polar. Now, small amounts of very small polar substances can go through there, like water, but not at a very rapid rate. So if we look here, the diagram on the right, we have some sort of lipid soluble solutes in the extracellular fluid, and they're able to move down their concentration gradient from high to lower concentrations. And then we have facilitated diffusion. Here we're going to have helpers. That's what facilitate is. So there's going to be some certain hydrophobic molecules, and I think this is incorrect, because if they were hydrophobic, they should be able to go right through the membrane. So these would be hydrophilic, those that can dissolve easily in water, which would be polar and ionic substances. Examples of polar molecules might be glucose, amino acids, and then there's ionic substances. Those can be transported passively down their concentration gradient, but we're going to need the help of proteins. And so there's two general categories here. Then there's the carriers and the channels. So we'll call this carrier-mediated facilitated diffusion. And then if there's channel proteins, then channel-mediated facilitated diffusion. And here they move through channels. So there is a difference between the type of protein. Looking at carriers, which is modeled in the image on the right, these carriers are going to be transmembrane integral proteins, which means they go into the membrane. Each of these carrier transports specific polar molecules such as sugars, amino acids that are too large for membrane channels. So we may have one that's specific for glucose, for example. So we have a glucose carrier, and it only binds to glucose. And so here the molecule, when it binds to the carrier, the carrier is going to grab on or envelop the solute. And when that happens, that causes the protein to change shape and move the molecule across the membrane down its concentration gradient. So here you can see we have higher concentration of these solute particles. And when they bind to your carrier protein, that carrier protein changes shape and allows the molecule to move through down its concentration gradient. So this does not require any energy. Now, there are some limits here. How fast this type of diffusion can occur. Carrier mediated diffusion depends on how many carriers you have for that solute, and they can become saturated if they're all working at the same time. So an analogy would be when you're trying to check out at a supermarket and there's like only one cashier, but a lot of shoppers are trying to check out, so that's limited. So if you want to speed up the process, you would have more cashiers. If you open up more cashiers, then we can pass more shoppers through, but eventually every one of the cashiers will be working, and again we hit a maximum speed at which we can move shoppers through there. Same idea here. Once every carrier is doing the work there, and you can't go any faster. So we could probably draw this, the rate here at which the diffusion would occur. And so we would say rate on this side here for the speed at which we're moving particles across the membrane. And so as we start increasing the concentration of the solute, whatever we're trying to move across the membrane, and the analogy is shoppers, the more shoppers you have, well, more shoppers, you're going to move more through the cashiers, more solute, you're going to move more through the membrane. But eventually all the cashiers are working at the same time and you can't move any faster. Same with the carriers. Eventually all of your carrier proteins are working at the same time, so you can't go any faster, and so we would level off at a certain concentration. So it would be at this rate here that we would hit saturation. So that would be at some concentration down here. And again, that rate depends on how many carriers you have. So now we have the second type of facilitated diffusion, which is the channel mediated here. And here the channels are going to have this pore that goes through the membrane here, and that the inside of that pore is going to be hydrophilic there, and so it would allow anything that's polar or hydrophilic to go through there. And so, and these can be specific, so you can have channels specific for, say, sodium. So it would be a sodium channel, for example. And some are maybe even be specific for water, which are called aquaporins. So there's some specificity here. And again, the key feature here is that whatever particles are selected to go through, if this is a sodium channel, it would be sodium moving down this concentration gradient. So you see you have your higher concentration on this side of the membrane, and over here we have low, so the general movement would be from the higher concentration to lower concentration, so it's passive. There are going to be two types of channels that are going to be important in understanding how cells function in different tissues. One is leakage channels, and they're always going to be open, so we're constantly having movement as long as there's a gradient. And then we have gated channels, and those gated channels will only allow diffusion when they're open, and so they can be either open or closed. If they're closed, then we won't have diffusion until they're open, and these are going to be controlled either by chemical or electrical signals. So if it's a chemical, it might be a little chemical in the area. When it binds to a certain area in a protein, like I'm drawing here, then that can cause that channel to either open or close. Or if it's electrical, then a change in voltage on the membrane. Membranes can have these membrane potentials or little slight voltages, and any changes in that voltage can cause the channel to open or close. And we have osmosis here. And osmosis, again, is the movement of a solvent, not the solute. And in this case, usually it's water, and we'll move across the membrane here. So water is going to diffuse across the membrane, just like any other solute would, from where water is more highly concentrated to where water is less highly concentrated. Now, water can move at a relatively slow rate through the membrane because it's a small molecule, but it's going to have some difficulty because remember it's polar, and we're trying to get through a non-polar region. So the speed at which water can move is greatly increased by very specific channels for water. Again, they are called aquaporins. Now, again, this is usually going to occur when there's a difference in the concentration, and that difference in concentration of water is due to differences actually in concentration of solutes on either side. So in order to better understand osmosis, we're going to define a term called osmolarity, which is a measure of the concentration of total solute particles. So it kind of sounds like molarity that we studied earlier, except for molarity, you're dealing with a very specific substance. For example, a 2 molar solution of sodium chloride. But in real cells, in real tissues, those tissues have many different solutes in there. So the number of particles is more important here of the mixture of particles. So when we have different osmolarities there, that's going to create different concentrations for the water itself. So water concentration will vary with the number of solute particles because those particles are going to displace water. So if you have more solute particles, I'm going to draw 2 beakers here, one with a lot of dots to represent solutes and one with few dots here. So in this case here we have a higher osmolarity here, but that means I'm going to put ASM for osmolarity, and here low osmolarity. Then this means we're going to have those particles are going to actually take the place of some of the water there. So that means there would be a relatively lower water concentration than the beaker on the right. This one would have relatively higher water concentration. So that's going to create a gradient not only for the solutes but for the water itself. So when solute concentration goes up, basically the water concentration goes down and vice versa. So that's kind of modeled here in simple pictures that I drew up there at the top. Now water is going to move by osmosis from areas of low solute. Remember, low solute means actually high water concentration, to areas of to high areas of solute concentration. And if you're high solute concentration, then you have low water concentration. So really water is moving the same way. So we can't get confused here, because oftentimes we refer to the solute, but it's a solvent that's moving. So when the solute is low, water is high. When the solute is high, water is low. So water is going to move down its concentration gradient. So we have to be careful with how these terms are used. So we're going to model this process with a U tube here, a literal tube shaped like a U here. And here we're going to see how the movement of particles goes if we were to place a barrier between the two sides of the tube. So here's a freely permeable membrane here, and it's going to be free to move for both water, which is the blue shaded area, and the solute particles to move. You can see there's different concentrations. So on the left, we have a lower osmolarity than on the right. But with a lower osmolarity on the left, that means we have a higher water concentration over here. And on the right side of the tube, we have a lower water concentration over here, even though it's a higher solute concentration or osmolarity. So regardless of the case, because this membrane moves, is going to allow both particles to move, then overall we're going to have more red particles moving to the left than to the right, and we're going to have more water moving to the right to the left. But eventually that movement or diffusion and osmosis that's occurring within this tube is going to occur until we hit equilibrium. So given enough time, then the particles are going to spread evenly, and now we're in equilibrium. But now if we change it, change the rules here, so we start with the same setup that we had earlier, but we instead place a selectively permeable membrane there in the middle that is only permeable to water, and so the solute particles cannot move down their concentration gradient. So no diffusion occurs, but osmosis, which is the movement of water down its concentration gradient, so it's diffusion of water basically, then we're going to have a net movement of water. So overall water is going to move to the right, and it's going to be moving to the left, but we're going to have more water moving to the right. Let me see if I can draw that here. So overall, I'm going to draw an arrow. We're going to have movement of water through that membrane to the right and some moving to the left, but because more is moving to the right, we're going to have a net movement of water. Since the solute cannot move through there, then once equilibrium is reached, in which the concentrations of the solute and the water are the same on both sides, we're going to see that overall a net movement of water occurred toward the right, and so the water level rose in that tube there. Again, equilibrium was hit, but only the water was able to move. Only osmosis occurred in this system. So the movement does involve some pressures when it comes to osmosis, and one of them is going to be hydrostatic pressure, and the other one is the osmotic pressure. And so we went back to that YouTube situation, and we had that situation where we had that selectively permeable membrane, and the water was allowed to move and more water moved to the right. If I had put a barrier right here and didn't allow water to rise up, as was the case that we saw in the tube on the right, and eventually the water stops right here, and the containment of the U-tube and that cap there at the end is going to build up this pressure that's going to prevent more water from moving. So the pressure of the walls of that container would be referred to as hydrostatic pressure. And that's going to counter the pressure due to the movement of water, or the solvent, or the osmotic pressure. So that would have been the inward pressure of the water moving into the right side there. So once those two pressures are balanced, then we have no more net movement of water. So osmosis would stop. So this is similarly modeled in cells. And so that hydrostatic pressure would be produced by the membrane of the cell itself. So that can provide some back pressure on there from the cell itself. And then the osmotic pressure is the inward pressure due to the tendency of water to be pulled into that cell when the cell has higher osmolarities or solute concentration. So the more solute inside the cell, the bigger the cell, or the bigger the pull of the water to enter the cell. And this can result in a higher osmotic pressure inside the cell. So again, this would occur until the two are balanced. So now how this plays out within cells really does depend on what the concentration is inside the cell of solutes and outside, or the osmolarity. Now, I already mentioned that when hydrostatic pressure is going to equal to the osmotic pressure, we'll have no more net movement of water, even though the concentration, solute concentration, may be different on either side. Once those pressures are balanced, then no more moving. Plants have a special feature. So even if they have high osmolarities inside and a lot of osmotic pressure, they're going to have a cell wall on the outside. And animals, and humans are animals as well, we don't have cell walls. So that cell wall, so they have a cell membrane, and then outside they have this cell wall. And that cell wall can help counter or provide that hydrostatic pressure. And so they won't have any problems with having too high osmotic pressure, because that hydrostatic pressure will counter that completely. But animal cells, if the difference in concentration is too great and the osmolarity is too high inside the cell, then we're going to have a greater osmotic pressure, and the cell will increase in volume, and eventually it can burst because there's no cell wall to provide that enough countering hydrostatic pressure to prevent any more further movement of water in. Now, water can also leave the cell, but you'd have to have a lower osmolarity in the cell. And so that would cause the osmosis to move in the opposite direction. This can cause the cell to shrink. And any changes in volume can disrupt function, because if you lose water from the cell, then your concentrations of electrolytes and other things are going to disrupt your metabolism in the cell. And then again, the problem in our cells is if too much pressure builds up in the cell, then you're going to have too much water coming in, and then that can cause the cells to burst. So this brings up a term then called tonicity. Tonicity is related to your osmolarity, but tonicity is the ability of a solution to change the shape or tone of a cell by altering the cell's internal water volume. The ideal situation would be to be in an isotonic solution, that is, the cell in an isotonic solution. And here, that cell's going to have the same osmolarity as inside the cell, so the volume remains unchanged. So I have that isotonic model here. And the green dots just represent the solute particles in general. The plasma membrane is in white. And so overall, those dots are drawn, those solute particles are drawn to say we have the same osmolarities on each side. So water will still move through the membrane, but we're going to have equal amount of water moving in as moving out. So there's going to be no change in volume. And then we have a hypertonic solution. And the hyper means more, and we're referring to, again, the inside of the cell when we compare to the outside. Okay, so it's a relative. You have to compare the cell. So if the cell is in a hypertonic solution, then you're going to have more solute particles outside, and that's modeled right over here. So there's a greater osmolarity outside the cell. So overall, that means there's lower water concentration outside, greater osmolarity, more solute particles, but lower water concentration overall. So here you're going to have water moving out, more water moving out, and you're going to have some moving in, but the water's moving more out. So there's going to be a net diffusion out. And what we have here is a loss of water overall. So the cell can shrink. And that shrinking can be referred to as crenation. And then there's the opposite of that, that's hypotonic solution. And there's going to be a lower osmolarity than inside the cell. And that's going to result in water, a net movement of water inside. So you can see here we only draw one dot outside the cell to show there's a lower osmolarity outside, which means higher water concentration. Inside the cell, you're going to have a greater osmolarity but lower water concentration. So overall, there's going to be that osmotic pressure going in, more water going in than going out. And so here, we're going to be gaining water. And if too much water goes in, then that cell can burst, and they call that lysing when the cell bursts. So here we see this modeled with red blood cells, and on the left is isotonic, so there's going to be no net gain of water there. Here's this stuff here. And those were the arrows I drew a while ago. And so in the left here, we have our isotonic solution here. And in that case, there's going to be the same movement inside and out, so no net movement of water. In the case of hypertonic solution, the osmolarity is greater outside, so there's going to be a tendency for more water to move out than to move in. So overall, the cell shrinks or crenates. And then in a hypotonic solution or environment, there's going to be a greater osmolarity inside the cells. So there's going to be a greater movement of water in and less coming out. So overall, we're going to gain water, and then we can lyse, which would be bad for the cell. So looking at osmolarity a little closer and how this certain different types of substances might be measured and how they influence. The osmolarities is going to be equal to the molarity times the number of particles when it comes to ionic substances here. And so it really depends, because if I were to have, so again, this is ionic, but if I were to have something like glucose, C6H12O6, it's a molecular compound, not ionic. And so if I have 1 mole of glucose, and then I go and I put it solid glucose, and then I go and I dissolve it in a solution, in a liter of solution, and now it's aqueous. So now I have it dissolved in water. So I'm put AQ. Now it's going to be 1, and I put it in 1 liter. Now it's going to be 1 mole per liter, or 1 molar glucose. And so 1 mole is going to give me 1 mole per liter. But for an ionic substance that breaks apart when you dissolve it, it can form more than the number of particles we started with. So a mole of sodium chloride, when we put it in a liter of water, it's going to break apart into 2 particles. So it's 1 mole of this is going to give us 1 mole of sodium particles and 1 mole of chloride particles. So that's now equal to 2 moles. Okay, so now we're going to have 1 mole dissolved in 1 liter. That's going to give us 2 moles per liter. So now we're talking about 2 osmoles here. So that's a difference there when we're talking about ionic particles. So a 1 molar solution of sodium chloride equals to 2 osmoles. So number of osmoles is just, we're talking about number of particles. And then if we took something like magnesium chloride, which has a magnesium and 2 chlorines, the formula unit for this substance, when you dissolve it, it's going to produce 1 mole of magnesium chloride is going to give us 1 mole of magnesium ions, but 2 moles of chloride ions. So 1 mole of this, or a 1 molar solution of this, is going to give us 3 osmoles per liter in solution. So the osmolarity then, when we express it, would be not moles per liter, but osmoles per liter to indicate total number of particles. So looking at a couple of practice questions here. Based on your knowledge of the cell membranes chemistry, where would steroid hormones most likely interact with their target cell? Is it A, through interactions with the integral membrane proteins? B, inside the target cell cytoplasm or nucleus? Is it C, steroid hormones don't interact with target cells? Or D, on the exterior surface of the cell? Next question, a red blood cell placed into a container of distilled water, just pure water with no solutes dissolved in it. So what will happen? So it's asking what will happen. A red blood cell placed in this distilled water will blank water via blank. So what will go in the first and second blank? Is it A, gain, and then the second blank diffuse? Is it B, gain, second blank osmosis? Is it C, lose, second blank diffusion? Or is it D, lose and second blank osmosis?